Okay, so let's get started. Hello everyone. Welcome to our today's museum seminar. Um, it's really my pleasure to welcome our today's speaker, Dr. Yongqiang from St. Jude. So just want to have a very brief introduction about Yongqiang. Yongqiang currently is associate member in Department of Immunology in St. Jude Children's Research Hospital. So Yongqiang actually completed his PhD training in Rutgers University in New Jersey in US, study cell biology and development biology, and then after he completed his PhD training, he moved to a Memorial Sloan Kettering Cancer Center, did his postdoc in immunology, and then did really remarkable and very productive PhD postdoc training. And then I think a lot of us probably already have learned Yongqiang's work from his work on the FOP3 Treg regulation. So after he finished his training with Sasha Rudensky in Sloan Kettering, he from 2016 he started his independent research program in St. Jude as an assistant member, and then get promoted as 2022 as associate member in St. Jude in the Department of Immunology. So Yongchang's lab actually mainly interested in T cell repertoire diversity and function, and then mainly aims to increase our understanding of how diverse antigen receptors of regulatory T cells are selected and sustained by genetic, epigenetic, and environment factors. Now, moreover, they also investigate how the magnitude of this diversity controls the robustness and levels of immune tolerance involved in autoimmune disease and anti-tumor immunity. Now, the very long-term objective in Yongqiang's lab is to identify and understand regulatory circuits involve nuclear programs regulating this process to advance our understanding, diagnosis, and the prevention of immunological disease and cancer. There's a couple of different aspects in Yongqiang's lab mainly interested in, which including they study epigenetic regulation of Treg induction and the lineage stability, and also they at the same time investigate a redefinition of the activity module of FOP3 protein governing versatile Treg function. So I think without further ado, I think Yongqiang gonna to tell us some of his latest work from his lab about Treg cells in cancer and cell tolerance. So I think without further ado, thank you so much, Yongqiang, with us being with us today, and we are very much looking forward to your talk.
Thanks, Tran, for your nice introduction. It's really a pleasure, and I appreciate your effort to organize such a meeting. I know it takes a lot of time. For us, we will try to be save our effort. Really appreciate. And today I'm gonna talk about something about regulatory T cells, which has been working for more than 15 years. I thought something come up to my mind recently, try to organize into a coherent understanding of T-Rex biology. And for those who are not familiar with T cell biology or immunology, I would like to emphasize the focus of our study. So immunologists hope to understand the fundamental questions, how to distinguish in the first place self versus pathogens. This is the initial definition of immunology, and we want to understand how we defend, fight against the pathogen. And later on, as a part of the side effect, we have to deal with how to distinguish self or distinguish harmful antigens such as food, commensal bacteria, from the real pathogens, such as various parasites, bacteria. And in addition, in recent years, there's additional understanding of immune system, how it functions in monitoring self organs such as stress damage. So this all belongs to a broader definition of immunology these days. Broadly speaking, immunology is going to monitor the integrity of tissue, whether the challenges come from pathogen or from infection or from foreign antigens, even though they are harmful, or to monitor tissue dysregulation. So this is a broader definition of immunology. So my aim today is try to provide a special angle to look at a subset of T cells called regulatory T cells, with the hope to offer your special perspective to look at Treg cells. As you know, the cells has been studied more than 20 years ago. Many things has been published. Well, with so many different aspects of T cell biology, I hope that we can have a simplified model to understand how the cells work fundamentally, and also hope to re-examine the functional fundamental principles, propose hypotheses, or predict additional important experiment for basic and translational research. This is the regulatory T cells. The fundamental role of the Treg cells was proposed initially to suppress conventional T cells that triggers inflammatory response to many antigens such as self versus non-self or including tumor. So Treg is going to suppress this process. The question has been intensively investigated in the field is how tolerance is induced regarding the target specificity or the level, and how tolerance memory, I try to like to call tolerance memory, is maintained or refreshed over a longer period of time. It's compared with uh, our brain or behavior memory. I will cover this topic briefly in this presentation, give kind of comparison and contrast. So our focus, my lab is focused on the fundamental principles, mechanisms governing immune tolerance, trying to understand what's the basic concept. I want to make things simple, given so many parameters, mechanisms. Can we have a simplified understanding, such as Treg induction linear stability or functional flexibility? I also hope to understand whether the conventional T cells and the Treg cells using different mechanisms, we call the polarity, polarized mechanisms to control their function or differentiation. And in recent years, over the last 5 years, we spent a lot of time to look at novel technologies. Can we introduce new methodologies for T cell research, including regulatory T cells? I also have a lot of interest try to promote the Treg function using synthetic biology approach with the goal that we are able to modulate the tissue microenvironment for basic research or translational applications. So today I'm going to focus on one project, mainly basic research I have been working on for more than 15 years, with the hope that it brings some fundamental understanding or maybe interesting model for you to consider. As I mentioned, the Treg cells was initially proposed to control conventional T cells. Is still the main topic I'm going to discuss today. So the cells we know with many mechanisms discovered, you probably can read from review papers. Treg can suppress the target conventional T cells in an antigen-specific manner. Essentially, has to be antigen-specific according to our definition of adaptive immunity. But the Treg cells also can suppress the cells if they are getting closer by. It's called bystander suppression. So the way to achieve antigen-specific suppression or bystander suppression, standard suppression, can be mediated by different mechanisms, as you can anticipate. For those you can find in any of the review papers. I don't want to spend too much on this topic. The consequence of the suppression is going to let T cells to avoid attack to self-antigens, commensal microbiome, food. We also want to take advantage of this to avoid allograft-induced immune response. And tumor is a different context. A tumor in the tumor settings, we want to avoid the tumor to take advantage of Treg suppression function, allow them to escape immune attack. And lastly, and in the recent years, Treg has been discovered that in addition to play an immunosuppressive role, also monitor tissue integrity as a consequence to uh, prevent the tissue damage or promote regeneration. No matter what specific aspect of the Treg play a role in the body, we want to understand what's the fundamental concept of this process. So to make this story simple, I want to summarize into such a simple curve. So over time, you can see the tolerance level increase or decrease. The black line try to show that tolerance is induced during Treg induction. This process going to allow the host us to uh, induce tolerance for particular antigens. Once induced, we ask the question how this memory, tolerance memory, is maintained over time. Are they going to decay? How quickly they are going to decay? In what context? So this is the question we hope we are able to address in our study. Treg induction is the first step, how this tolerance mechanism is induced. A lot of work has been proposed. Here shows the how Treg induced in thymus. The story is relatively simple. In fact, probably is not that case. So to simplify this process, so the antigens, self-antigens can be induced in thymic epithelial cells located in a medulla called the medulla thymic epithelial cell (MTEC). The gene that promotes self-antigen expression called AIR. To simplify, that self-antigen being induced and presented to T cells during differentiation. Those that induce stronger TCR signaling will go to apoptosis. In the middle, they will induce Treg. If they are weak, they will become conventional T cells. This is what we understand so far. And the signals that promote Treg induction include something self-antigen, food antigens. The signal including IL-2, TGF-beta, and additional modifications such as vitamin C, vitamin A, short-chain fatty acid. And this process is suppressed by pro-inflammatory cytokines. I'm not going to dig into the specific details, but I would like to highlight the fundamental concept, how the body, the immune system is going to distinguish self, non-self, or good versus bad antigens. So this, for self-antigens, will be relatively easy to understand, those that T cells exposed during thymic T cell differentiation. In that context, system seems don't need to distinguish self versus non-self, such as food and commensal bacteria, because they don't reach, most cases, don't reach thymic epithelial cells. This is relatively simple. In fact, actually, how the individual antigens being expressed seems to have a lot of things to explore. We don't know for sure yet how this antigen can be induced in thymic epithelial cells. They are come together often with those antigens potentially induce inflammation. The question is how the system are able to distinguish. Recent work showing that a population of dendritic cells expressing R-gamma T initially was considered innate immune cells, was able to induce tolerance. However, many things remain bizarre in this process. Personally, I don't understand yet how come a system are able to distinguish good versus bad antigens. For those good ones, We want to induce Treg cells and immune tolerance memory. We want to understand what's the given somatic complexities, what's the fundamental thing that governs Treg induction and tolerance level. And for this, we have to mention that the key factors control Treg fate and the suppressive function is a nuclear protein called FOSP3, which is a key protein discovered 20-some years ago. Without the FOSP3 protein, cells will turn to something like a conventional T cells. They are pro-inflammatory. With this protein, human or mice develop systemic lethal autoimmunity. I'm not going to dig into this. This is well known. But I would like to take advantage of FOSP3 to understand how Treg are induced, because FOSP3 makes the system relatively simple to study. By turn on or turn off FOSP3, give us a convenience to look at the Treg fate determination. Here I want to show you how we visualize self-antigen or foreign antigens and the relevant relationship with Treg and conventional T cells. Imagine in the body, we have millions, if not billions, of different antigens. And for individual antigens, as multiple T cells are able to recognize. Here is on the give such a two-dimension diversity of antigens. Each antigen has many epitopes. Each square, see, you can see the Treg or conventional T cells. They are present at different frequency. We can often for Treg studies, we don't know what they react to, given such diversities. And the TCR also so diverse and covers probably more than 100 millions of different possible combinations. So this question has been raised since the discovery of Treg cells. The study has done by Qi in Washington University, St. Louis. He actually did an experiment trying to measure how particular T cell, transgenic TCRs, how they become Treg. So he cloned the TCR from Treg cells, put it in the transgenic mice, measure how frequent they are going to become Treg. He titrated this using mixed bone marrow chimeral setting. He found that when you deliver more T cells, doesn't mean that they're going to become Treg. And after all these calculations, he found that about for a particular TCR transgenic T cells, there will be 10,000 cells will become Treg. The conclusion is that the self-antigen is limited. So that you're actually missing antigens, even though you produce a lot of transgenic T cells. So only a small fraction of the cells become Treg. Not to mention that some of the TCRs actually recognize multiple antigens. It's a general problem for T cells. We call it cross-reactivity. And Mark Jenkins did another kind of experiment. He measures for particular antigen how the system is going to react, induce Treg or induce different kind of tolerance. He measured the reactivity. You can see a broad spectrum of conventional T cells against a different kind of GIP, but nonetheless the same GIP but different, driven by different promoters. It's interesting that the way that you deliver GIP make a lot of differences in the selection. Many of the selection probably go through negative selection. He further measured the Treg frequency compared with the conventional T cells, such as TFH, TH1, TH17 cells. The conclusion is that there's a broad spectrum of selection. Sometimes GIP can induce negative selection, sometimes induce Treg cells. The range of the Treg cells can come from a few, maybe up to a few hundred cells. The question raises what determines the induction efficiency or selection efficiency? What if there's a mechanism impairs in efficiency? What's the consequence could be? So I put this into the context of billions of different TCRs or millions of antigens. How can we understand the basic diversity Treg target coverage and the tolerance level? So this has been challenging. We take a different approach. We look at the FOSP3 gene control regulation because, as I said, the FOSP3 offers such a convenience for you to control Treg fate by a single genetic switch. So we look at the enhancers that control Treg effect. I'm not going to dig into individual pathways that enhancer respond to. I would like to highlight that the several enhancer controls Treg induction. One of the enhancer name was given by a former colleague called CNS3, which is poised by epigenetic modifications H3K4 monomethylation. Without this enhancer, you see a reduced Treg induction by about 30%. But otherwise, you look at the mice, there's perfect. There's nothing wrong. You challenge the mice. I spent years look at the mice, nothing wrong with this mice. And so my career would be trashed if I keep doing this. Uh, Even though that we can see that the Treg diversity was reduced slightly. Nonetheless, the system were able to tolerate such perturbation. I was disappointed, maybe not surprising, as a biological system, we have to deal with kind of perturbation variation. But the question is how the system is going to cover Treg, cover the antigen specificity or determines the threshold of tolerance. So this gives another idea we tested by combining CNS3 knockout versus with the air deficiency. As I mentioned, air promotes self-antigen presentation. Otherwise, it's also surprising that without air alone, animals actually looks okay until they are about 3 or 4 months old. So the mice can breed for a few months and then start to develop a systemic autoimmunity. As you can see, the system almost can also tolerate kind of air deficiency. But when you combine these two systems together with the mice lacking both, they quickly develop severe lethal autoimmunity. We can see this change within 2 months. You can see autoantibody react to many organs, including intestine, skin, retina. What this study suggests that there seems synthetic interactions that perturbation of one part of Treg development, tolerance and induction, same system can handle pretty well. But if you perturb both, you break down the tolerance to a level that leads to a severe autoimmunity. In the meantime, we look at the different enhancers in the locus. One named by Shimon Sakaguchi called CNS0. This element was responsible for chromatin binding protein SANT-B1, and the CNS0 also controlled by the histone methyltransferases MIL4. Without this particular enhancer, cells this actually is a ChIP-seq data to measure the responsible element toward L2 stimulation. So this is a plot showing that the CNS0 responds to L2 and STAT5. Without CNS0, you see a similar reduction in Treg development, but otherwise the mass also perfect without any perturbations. Even though that TCR alpha chain or beta chain was reduced by the diversity. We combine, again, use a similar strategy. We found that the combination of CNS0, that time we gave a different name, but otherwise was the same enhancer. In the absence of both CNS0 or CNS4 with air, we saw a severe autoimmunity and reduced tumor growth if you inoculate tumor into this mice. So this is basically generate a broad spectrum of Treg induction. We actually further push this understanding, see whether enhancer was able to actually coordinate in somehow. To test this idea, a former postdoc in my lab did a CRISPR TALEN screening to look at the potential interaction between enhancers using CRISPR. And she found that indeed the enhancer CN0 and CN3 potentially interact. And when both enhancers was knocked out, you can see the mice quickly die of severe autoimmunity, was almost comparable to FOXP3 deficiency. And this is the induction efficiency, this is the diversity quantified by TCR sequencing. So this, to summarize all the work we did in the past more than 13 years, to summarize everything into such a diagram, we found that deletion of individual elements, CNS0, CN3, or air, actually reduce subtle change in the Treg induction efficiency, but otherwise the animals were able to tolerate really well. We further reduce the Treg development through combined deficiencies. Now we push the system further down, lead to severe autoimmunity, and eventually kill the animals in mice with around 2 months of age. So our work was done in a polyclonal context without knowing specific antigens. The question will be how exactly the system works. We proposed several models trying to understand this, since none of this actually was good enough. So there's a work recently was published by Peter Savage's group. He actually has been looking at the antigen-specific Treg for more than 20 years. And he generated the mice lacking prostate-specific peptide, so that there's particular Treg cells will be missing, but otherwise the animals are okay. He didn't notice any dysregulation in these animals, even though that this particular prostate-specific Treg cells are missing. He deleted in the antigen presentation in thymic epithelial cells. So the best way to explain this data is that appears to be that Treg are able to provide a bystander suppression, even though some more clones of Tregs are missing. Well, he further pushed the system by delivering more antigens using listeria. By making transgenic listeria, he can deliver high dose of prostate antigens. In this case, without this particular antigen-specific Treg cells, there will be severe inflammation in prostate. When he provides specific Treg cells, this inflammation can be fully suppressed. So this suggests that there will be an antigen-specific suppression required when antigen is presented at a higher dose. So from here, we try to summarize that our studies of polyclonal Treg cells without knowing specific antigens point to a probability of Treg induction efficiency. We call the stochastic Treg induction. Means that for particular antigens, there will be more Treg or less Treg cells. There will be a considerable variation across the spectrum. So while the system can handle this, even there will be particular perturbations, such as particular enhancer deletion or error deficiency, we think that the bystander suppression can fully tolerate such kind of variations. However, if the antigens are presented at a higher dose relatively compared with the Treg cells, now you need the antigen-specific suppression. To put the story simple, we think about the conventional T cells, how the cells are going to win different signals. Part of the signals come from antigen-specific stimulation, such as TCR signaling or co-stimulation. Part of the signals is negative signals from Treg suppression. The conventional T cells are going to win the two signals. A positive signal from TCR stimulation, co-stimulation, and negative signals from Treg-based bystander suppression or antigen-specific suppression. So now put in this spectrum, with the antigen load versus Treg ratio is kind of low. Now bystander suppression mechanism is able to suppress conventional T cells because during this time, TCR stimulation, co-stimulation is relatively weak. However, with antigen load is relatively high, conventional T cells now need a stronger suppression in order to keep their tolerance state. So now antigen-specific suppression become a dominant player. So the relativity of the antigen load now determines the suppression mechanisms, how the system is going to be involved in order to maintain tolerance. To further titrate up the Treg load, I think there will be a broad spectrum of Treg versus the antigen ratios. So that to further understand this, I try to introduce relativity. I know it's not, we are not a physicist, but the idea is similar. So everything about the relativity, right? Relativity of the antigen load versus Treg or Treg versus conventional T cells In this context, it's kind of interesting that when the antigen load is low, Treg bystander suppression can fully control conventional T cells, giving immune tolerance. While antigen further increase, now need the antigen-specific suppression. Normally, systems are okay, we can handle this. While particular self-antigen are expressed on higher levels, our antigen-specific Treg cells are able to suppress those conventional T cells, we are fine. But if you further increase the antigen load, such as particular tissue damage, that lead to the release of many of the self-antigens. Now you titrate out the Treg suppression. In this case, conventional T cells are going to release the tolerance, lead to the inflammation. So when you look at this curve, now we can kind of have a simplified model to understand the relativity of the antigen load, conventional T cells, Treg number, or coverage. So this model makes a lot of predictions, given the relative ratios of all the three key players, not to mention that maybe additional players also participate. But given this, we can make predictions, for example, in the connections between bystander suppression and antigen-specific suppression, because these two parameters are often connected. And given the complexity of the Treg function, always relies on TCR signaling. So we think that in vivo, even for tissue protection function of Treg cells, the Treg cell need a charge by specific antigen stimulation, like a EV. We are driving these days. You need to charge your car by antigen-specific stimulation. Once you got enough power, you can drive for 100 miles. When you run out of juice, electricity, you have to come back to the charging station to charge your car again. I think this offers an interesting model to test in the coming years. In addition, that we think that by planar antigen-specific suppression or bystander suppression, we can offer some guidance for Treg-based therapeutic treatment of such as autoimmunity. Next question I raised is how the memory, once formed, how is this going to be sustained over time? This goes to another question to be asking for 20 years is how stable Treg fate really is in vivo. A study performed by Sasha Rudiskin and former trainees using cell fate tracing experiment showing that Treg can maintain cell fate for several months without a significant loss in a steady state or after several challenges. But indeed, you still are able to see some of these Treg cells potentially lost the FOXP3, become X-Treg cells. These cells are able to promote inflammation. The question has been in the field is whether how Treg indeed is able to lose the fate, in what context, what determines this stability or robustness of the Treg-based tolerance, and how this system is able to sustain their fate in the presence of perturbations or reprogramming. To answer this question, I think we should go back to the inheritance of a cell fate. This is a fundamental question for epigenetic regulation of gene expression. This model is complicated here, proposed by Dan Rimberger several years ago. But I want to bring your attention to the simple questions with cell divide. How are two daughter cells able to maintain their fate? Well, many epigenetic mechanisms are going to dilute out during this process. So I think all Treg cells offer such an interesting system to answer such a fundamental biochemical question. The key point for Treg cells to address this question is because Treg appears to be long-lived in animals. The cell tracing experiment indicates that Treg cells in mice can survive at least several months. This is corresponding to several decades in human beings. So the question is how cells can maintain their fate over such a long period of time. And this goes to the epigenetics. Epigenetics offers such a biochemical basis for us to understand cell fate inheritance, because we speculate that the cell fate has to be maintained by such kind of marks, and they are going to remember what state they are. So this search has been done 20 years ago. End up found that demethylation seems fit with such expectation. Here you can see that Treg cells actually has extensive demethylation in, in, in a Treg-specific manner around the phosphate locus. This also happens to many Treg functional genes. In vitro, that when you induce the Treg cells, when you provide an activator of the demethylase, vitamin C or ascorbate, as in this case, you actually can achieve such kind of epigenetic change. The element controls this particular DNA demethylation is enhancer CNS2. We delete the CNS2, we see that after cell proliferation, they can easily lose phosphate 3, especially in the dividing cells. So the fit with our expectation that enhancer linked to epigenetic memory is going to contribute to Treg instability. And the mice without the CNS over several months, you can start to see that chronic inflammation occurs in some of the non-lymphoid organs. Upon challenge by, for example, LCMV infection or the multiple sclerosis, you see the similar consequence. So that's everything fit with the expectation of such epigenetic changes. But we were not happy with whether indeed this is the case, because without the CNS2, Treg still largely maintains their fate over time. So it appears to be something else also play a role. It's not dark matter. It must be something that in the locus controls the cell fate. For this purpose, we did a CRISPR screening, TALEN screening again. Now we found that the enhancer control induction also potentially play a role here. The enhancer called CNS0. And we did a double deficiency of CNS0 and CNS2. Now we can quickly reduce the cell animal lifespan to about 2 or 3 months. And the mice show large spleen and systemic autoimmunity. And the Treg cells will transfer looking at stability, they quickly lost the avidity. So these two enhancers appears to be able to control Treg fate. And we measure the cell proliferation TCR clonality, demonstrating that without these two enhancers, Treg cells won't be able to maintain cell identity during cell division, because without these two enhancers, we don't see many dividing Treg cells. So here, bring your attention to the mechanism that control Treg fate, not simply by a single epigenetic change of demethylation and phosphate enhancer CNS2, but also relies on another enhancers which has not been linked to epigenetic such as DNA demethylation in this context. So we propose that there's a multi-level cross-regulation of a cell fate through epigenetic memory and another pathway such as L2 STAN5, which always are there in Treg cells. So it's not simply DNA demethylation through an enhancer give Treg such a durable suppression through stable cell fate, but rather relies on multiple systems. So this goes to the curve again. When Treg are induced, we start to build a tolerance memory, but over time, there's mechanism involved to maintain such a memory through methylation, DNA demethylation, or through a signaling pathway that Treg cells always relies on. L2 signal is essential for Treg function. But what we don't know is how the system is able to maintain in different contexts. I draw the 3 different lines. Green line will be a stable fit, as you see in the normal tracing experiment. Blue or red has different levels of perturbation. And I don't have time today to really bring this into a immunological or normal physiological context. Indeed, actually, now we see that this can happen in different levels, depending on the challenges. Now we are able to trace this in a more relevant context. In the last few minutes, I want to bring your attention to the phosphate3 protein itself again. And when we talk about the phosphate3, we always think that the FOSP3 is a DNA binding protein or called a transcription factor that bind to DNA, determine target specificity. This idea has been proposed and has been thought for more than 20 years. We never challenge whether FOSP3 is doing something else. But look at the Treg cells. As I mentioned, the Treg is doing many jobs. Right? Treg can be resting, can be more active, more suppressive, or can do tissue repair functions depending on the context. So how this is going to be achieved? We will look at Treg cells as a sensor. Cells are going to respond to a tissue challenge for cytokines, antigen stimulation, nutrition state, oxygen level. So all this feed into Treg cells and that determines the cell response to this kind of signals. And as a result, Treg cells going to secrete cytokines or suck more IL-2. Cytokines such as IL-10 or IL-35 potentially are able to suppress immune response. Tregs also are able to secrete such as alpha-regulin. AREG is able to promote the tissue regeneration. And the target of the cells also are diverse. So Treg cells are able to do this kind of so diverse functions. The question has been there in my mind for more than 10 years, how Treg can do this. Is this through you specialized regulatory network or a phospholipid can do something in this process? This is the question we'll be trying to answer. Phospholipid protein is not new to us. It has been looked at very carefully from different angles and summarize here. Phospholipid can bind with more than 300 proteins, can be modified by different post-translational modifications. And then we'll be asking how phospholipid works in the context of gene regulation. The simple experiment we did several years ago is to look at phospholipid, exact phospholipid, how it bound to chromatin, using that time was a newly developed card-RON sequencing. We look at the binding in Treg cells where they are resting or after activation. Now we see something really striking that they bind to the chromatin, phospholipid bind to chromatin differently when cells become activated. We look at the binding site, we look at the genes that correlate with the binding. Some are going up, some are going down. I think this correlation meet with the changes of the Treg expression function. This is not trivial to me because we'll be thinking about Treg cells, as I mentioned, as a DNA binding protein determines target specificity. We assume that this is not going to change. Treg fate is fixed. Once the phospholipid is expressed, you turn on the switch, they become Treg, they're going to suppress. But in fact, that this appears to be context dependent. We further demonstrate that when you hit the cells with a cytokine, with TCR antigen, you can change this binding, meet with the motifs. And when you inhibit the TCR signal using cyclosporine, you can eliminate phospholipid chromatin association. When you look at the tumor, in the tumor environment, you see different pattern, different motif enrichment. And we further look at the chromatin dynamics using a proteomic approach. In this case, you can label the phospholipid complex in situ in live Treg cells and pull out the protein and label it by Beltin. We can purify this. You can look at the total protein bound to the phospholipid associated chromatin. Now you can see that signaling from after L2 stimulation or TCR stimulation, you can see dynamic association of the proteins within the phospholipid proximity within in situ settings. Now we can see the phospholipid appears to form a dynamic complex with the downstream effector molecules. We're using L2 signal or TCR signaling as two examples. We believe this is going to happen to other signal events. As I mentioned, the phospholipid has been shown bound with more than 300 proteins, and a lot of work has been done. Also, in vitro data has pointed to the phospholipid binding with DNA. Structure has been shown, sequence has been identified. Some has proposed that phosphate bind to microsatellite DNA. But our data suggesting a different way to understand the complex, how to interpret these complex interactions between phosphate3 and the associated proteins. This is a question we'll be thinking for years. And in this complex, something bizarre happening is that phosphate3 bind to DNA, according to crystal structure, appears to be really specific. But the associated proteins like NFAT, AP1 proteins, and NF-kappa B, they also bind to DNA. The question is who decide? If all bind to DNA, who decide the binding specificity? Or all of them decide. So our in vitro data, in vivo cut-around data suggest that the specificity is not determined by phosphate itself, rather it's determined by the associated factor such as NFAT or AP1 proteins. What's the big deal of this? I think this gives a way to understand how the complex regulate the gene expression in the context of the flexible Treg functions. We think we have a better understanding how this complex is going to function as a sensor of the environment. Let's see the TCR or L2 signal or other like hypoxic conditions are going to trigger these factors. Once they enter nucleus, they're going to form complex phosphate3, drive phosphate3 to the gene of interest to regulate gene expression in a context-dependent manner. So this gives us such a kind of flexibility to understand how the complex forms and how the cell is going to respond to environmental cues. That also raises many questions. I also call this relativity. Relativity of the numbers, numbers of the proteins, how they're going to, which one's dominant, which one is overwhelming in some time. So it gives cells to determine whether it's a pro-inflammatory condition or actually anti-inflammatory condition. So the complex gives a lot of clues to understand how cells are being controlled at this level. And we hope that by looking at this complex from this perspective, will give us some more insight to dissect the fundamental principles, how the complex works. Well, we look at the phosphate3. I'll talk to many people work on other TFs in T cells or maybe in other field. This is probably not new for phosphate3 itself. They could apply to other nuclear factors. I think the general idea would be that for these DNA binding proteins, they may not determine target specificity all by itself, but rather it's determined by multivalent interactions of the complex with DNA. With such multivalent interactions, what determines that time for that particular context and the sequence specificity, I think we need to look at individually for the context. In some context, maybe one part play a major role. Other contexts we may shift. I think this raises the questions for fundamental mechanisms, how the TFs control target specificity and gene regulation. Okay, I think I have a few more minutes. Here I want to briefly summarize what I presented today is some fundamental mechanisms determines how the Treg-based immune tolerance memory is formed in the polyclonal manner by determining induction efficiency and potentially target coverage. Or coverage also links to the representation. Representation is a factor determines the level of tolerance. So this also linked. And once the tolerance is formed, how this tolerance memory can be carried on for the longer period of time, because as our memory brain also remembers what we expect, what we met in the past days or years. So they are comparable in many ways. For example, if the information we processed 10 years ago will be useful, we may need this kind of information again, refresh memory. Same thing may apply to the antigens we are supposed to tolerate, but this antigen, if they come in, occasionally they're going to refresh tolerance memory. But if they were absent for some time, this memory will decay, as if we forget some stuff. I belong to that kind of person that I can easily forget, be a meaning that I'm ready to take more information, new information. So that's a way to think about tolerance memory, how to maintain the memory, how to refresh in different contexts, will be more meaningful for biological system, as we know that the memory was designed for prediction, right? Memory is going to predict what the system needs to deal with when they meet something again, right? Treg cells, if they remember an antigen, they're going to decide next time I meet this same antigen, should I fight or should I tolerate? Same thing with our memory, neuronal memory. We decide what we are going to do next time, we will see the same thing. And lastly is the fundamental drivers of the Treg function through phosphate protein. The protein appears to be complex, but there's fundamental principles we think we have some insights we can be used for further explorations or get additional experiment design. All these studies hopefully can help us to design experimental test in the coming years, potentially also help us to design strategies to engineer better Treg cells for therapeutic purpose. This is the last slide to thank the people who did the job here, and thank for the funding from St. Jude and NIH grants. And we hope that we'll welcome more people to join us to pursue pioneer research in Treg biology. Broadly speaking, covers the beyond stability. There could be stamina, aging, engineering, or technology development. A lot of exciting things are ongoing. I'll stop here. I'm not sure if we are able to answer questions